Hello, everyone. Welcome to our third annual IUFRO webinar series in celebration of International Day of Women and Girls in Science. This two-day event is starting today and consists of 3 sessions. My name is Devrim Semizer Kaming. I'm a researcher at the Bavarian Office for Forest Genetics in Germany, and I'm privileged to host this session on behalf of our webinar organization team. The event brings together an international panel of brilliant women researchers working in various fields of forest sciences. But before we begin, let me introduce you IUFRO. The International Union of Forest Research Organizations is a nonprofit and non-governmental international network of forest scientists. It promotes global cooperation in forest-related research and enhances the understanding of the ecological, economic, and social aspects of forests. Established in 1892 in Germany, IUFRO unites around 630 member organizations and thousands of researchers in over 115 countries. IUFRO's scientific activities in key forest research fields are spread over 9 divisions. Within the divisions, research groups and working parties address specific research topics such as silviculture, genetics, and forest health. This webinar is coordinated by Division 7, Forest Health. Forest Health Division is organized into 2 units, pathology and entomology, focusing on various aspects of forest health-related, research-related diseases, pathogens, insects, and environmental pollution. To learn more about the divisions or IUFRO in general, please visit our website at www.iufro.org. There you will also find information on upcoming events, publications, and how to get involved in our global network. So, I would like to now, acknowledge and thank each member of our webinar organization team. Their efforts made it possible to reach out to you today. And before we start, I will go through a few housekeeping rules. Our moderator will leave her camera on, so it feels lively for our speakers, but please leave your cameras off to preserve bandwidth. You're welcome to use the Q&A section to ask questions to our speakers. Our moderator will ask them to our speakers on your behalf. You're also welcome to use the chat for any other communication, such as connectivity or sound issues, or to say hi to your colleagues. Closed captioning is available in this meeting. You may activate them by clicking the captions button at the bottom right corner of your screen. If it's not visible yet, you may have to click on More first. During the webinar, we will send all participants a link to a survey on the themes related to the experience of women in forest sciences field. We would very much appreciate your anonymous contributions, and all genders are welcome. Our moderator for this session is Dr. Jessa Atta from the University of the Philippines, Los Baños. Jessa, now, floor is yours.
Thanks, Devrim. Hello, everyone. My name is Jessa Atta from the University of the Philippines, Los Baños, and I'll be moderating today's session. We've got an excellent lineup of speakers for you today, women scientists who are trailblazers in forest sciences. Each speaker will have 12 minutes for their presentation, followed by a 3-minute Q&A with the audience. After the 2 presentations, we will pause for a 5 to 10-minute break, which would be a good opportunity to complete the survey we posted earlier. After the break, the other 2 speakers will then continue. Again, welcome everybody to the first session of the 3rd IUFRO webinar series on women in science. Our first presentation will be delivered by 2 speakers. One is Dr. Rattiya S. Lippe. She is a senior scientist at Thunen Institute of International Forestry and Forest Economics in Hamburg, Germany. Her current research focuses on employment and labor market issues in the forest sector and related economic activities, including the informal economy, labor statistics, working and employment conditions, gender equality, and governance in the world of work. She will be co-presenting the talk with Yonca Gürbüzer. Yonca is an economist and statistician at the Food and Agriculture Organization of the United Nations in Rome. In her role, she serves as a focal point for gender and women's access to agricultural land and works on employment statistics and statistical methodology related to sectoral employment. Dr. Rattiya and Yonca, the floor is yours.
Thank you. We will share the screen and I will first give the floor to my colleague. You see my screen well?
Yes.
Perfect.
Thank you very much, Rattiya, and thank you very much, Jessa and colleagues for organizing this session. So good morning and afternoon, everyone, and thank you for joining us in this webinar. My name is Yonca, and today we are presenting the work carried out by FAO, ILO, and the Union Institute. In the past couple of years, we've been working together to quantify how many people work in the forest sector, who are they, and how employment patterns differ between women and men around the world. And today I'll walk you through the data, methods, and modeling behind the first global estimates of gender-disaggregated data on forest sector employment before I give the floor to my colleague Rattiya, who will present you the results. Next slide, please, Rattiya. Okay, let me start why forest sector employment data is important, especially when broken down by gender. First, the number of people employed in the forest sector is one of the most tangible and long-standing measures of socioeconomic importance derived from forests. It gives us a clear sense of how forests contribute to livelihoods and national economies. Second, this data play also has an essential role in evidence-based policymaking. Employment is directly linked to SDG 8 on decent work and growth. Having, of course, reliable data helps countries track progress towards this goal. In addition, time series employment data allow us to identify regional labor market trends and emerging opportunities. And the gender-disaggregated employment data go a step further, and we gain insights into occupational segregation and the changing role of women in the forest workforce. This understanding supports more inclusive and equitable employment strategies across the sector. Next slide, please. As I mentioned, the results we are presenting today come from a joint effort between FAO, ILO, and the Union Institute. Using this collaboration, we produce global, regional, and country-level estimates. These are now published in Global Forest Resources Assessment. According to the FRA 2025, the forest sector employed at least 42 million people worldwide in 2022. And in the coming few slides, I will focus on how this number was derived, the data, definitions, and methods that underpin this estimate. Next slide, please. Next one. Okay, our first step is to clearly define the frame. Who are we talking about when we say employed in the forest sector and who is included? To do so, we use the International Standard Classification, so-called ISIC, at the 2-digit level codes, and accordingly, we have 3 subsectors that we are covering with our estimates under forestry sector. The first one is the forestry and logging. The second one is the manufacture of food and products of food. And the third one is the manufacture of paper and paper products. Next slide, please. To estimate employment, we use the international definitions set by the International Conference of Labor Statisticians, so-called also ICLS, and according to which employment includes paid employment as well as the people who are in self-employment. We focus specifically on employment in a person's main job. So as well, the secondary jobs are excluded. Our primary data source is the ILO stat, which provides harmonized data on employment by sex and economic activity. From that database, we extract the ISIC level 1, ISIC level 2 digit codes, and the model estimates that feed in our model that I will talk to you in the coming slide. Next slide, please. Okay, the challenge is that we know that employment data is far from being complete for all countries and years. Many reporting countries have gaps across years 2011 and 2022, for which we produce estimates that you see on the left chart. And many countries do not have any data at all between these years. So that's also shown on the right chart in the screen. This missing data patterns, of course, creates significant challenges to come with a global number, and our aim is to address them in our modeling approach. Next slide, please. To overcome these data gaps, we developed the forest employment model, so-called FEM model. The idea is basically straightforward. We use observed employment data together with country characteristics, such as socioeconomic indicators, labor supply, and forest-related production to predict employment shares by subsector and by gender. But there are some challenges. The first one is the missingness of the data is not random. So countries with lower GDP per capita, smaller populations, or weaker labor force surveys are less likely to report any data. We correct this using sample selection weights derived from a logit regression that models the probability of data availability. Second, we run a weighted panel regression for each subsector that we showed you, for men and women, and for reporting and non-reporting countries separately. At the end, we use Lippe cross-validation to identify what is the best fitting model for each case. Finally, this modeled employment shares allow us to fill all missing data points and then adjust estimates back to observed values where available to ensure consistency over time. This process is what enables us to produce complete and comparable annual employment estimates for 182 countries. With this, I will give the floor to my colleague Rattiya. Thank you very much.
Thank you very much, Yonca. And now I will move on to some of the key results that we would like to share with you. But before I start, I just want to get a feedback. Do you see the slide title, right? Because
Yes.
Okay, perfect. Good. So the first thing I would like to mention that the FEM model already provide the global estimate, and based on the FEM model is, as Yonca already mentioned from her first slide, at least 42 million people worldwide are employed in the forest sector, which is about 1.2% of the global labor force. Out of these 42 million, based on our estimation, about 11 million women were employed in the forest sector. It's representing about 25% of the total forest sector employment. You can see from the bar chart here that the women employed in the sector remain relatively more or less the same in the past 20 years. This slide shows you the trend of forest sector employment disaggregated by male and female expressed as the relative share of the total workforce from 2011 to 2022 across region. On the global scale, you can see that 0.8% of women and 1.5% of men were employed in the forest sector. The gender gap or the gender employment gap persists over time and in all regions, which in particular, Europe has the widest gap with the male forest sector employment 4 times higher than those of the female. However, when you look between subsectors, you can see that the trend in female employment and gender gap appear to be more pronounced across subsectors. Let us start with the forestry and logging subsector. These charts show you the employment trend and the gender gap in forestry and logging expressed as the relative share of the total agricultural employment. On the global scale, the gender gap in forestry and logging is about 0.7 percentage point. In Africa, America, and Asia, the gap remains relatively narrow. However, in Europe, although the female employment in this subsector remains very stable, but it shows the largest gender employment gap at about 6.8 percentage point, followed by Oceania. Moving to or turn to the wood manufacturing subsector. Compared to the previous subsector of forestry and logging, the gender gap in wood manufacturing subsector is relatively larger on the global scale. Looking in Africa, for example, is experienced the notable growth of female employment in this subsector, but nevertheless, the gender employment gap in Africa is still the widest compared to the other compared to the other regions. The gap is about 7 percentage points. For the case of Asia and America, it shows that there's a gradual increase of the female employment in wood manufacturing, which suggesting that this industry is become more accessible for women. The third subsector that we consider in our analysis is pulp and paper manufacturing. We can see that the women participation in pulp and paper manufacturing remain low on the global scale. And also compared to the other subsector, there is a smallest of the gender employment gap in this subsector. For the case of Africa, for example, the female employment is declining. At the end of 2020-22, we found that only 0.9% of the total women who are employed in the pulp and paper manufacturing. But then compared to the region of America and Asia, the female employment in this subsector had a little bit increase from 2011 to 2020-22. For the case of Europe, when you compare, if you remember, for the case of forestry and logging, the gap is very wide. But for the case of pulp and paper manufacturing, both female and male employment in this subsector remain quite stable, but also maintain the comparatively small gender gap over the past decade. So in conclusion, gender disaggregate and sector-specific employment data are essential for assessing social, economic, and sustainability impact, and especially how the forest contribute to the job creation. However, based on our study, we can see that there's still a significant data gap across the country, especially in the developing regions. The FEM model that we developed already provides the first global estimate of women participation in the primary forest sector. This already helped to address cross-country data. But to move forward, we can also increase our accuracy of the estimation by enhancing or expanding the observed data in finer industry level, and also we can also improve the measurement of the non-wood forest product, informal work, and related sectors such as furniture, transportation, and ecosystem. By this, we will be able to fully capture the employment dynamic in the forest sector. And at the end, we need to move beyond the head count, because it's very crucial to understand what kind of job quality occurs in the forest sector, the safety, the social protection among the workforce, the working hours, etc. To have this kind of comparable data is depend very much on the sustainable effort in the international collaboration, stronger national statistical system, and also to improve the labor force survey. Thank you very much, and I would like to end our presentation on this slide. I will stop sharing the screen.
Awesome.
And back to you, please.
Thank you. All right, thank you so much, Dr. Rattiya and Yonca, for that insightful presentation. We now welcome questions from the audience. And I can see there is one here in our chat box. Are there any political or regulatory efforts to fill the gender gap in those sectors who employ less women in Europe? Any, either Rattiya or Yonca could?
Well, it's a very good question. I may not be able to fulfill this question, I have to say, because first of all, the study as of today is first to understand actually how many women employed in the sector. But from the experience that so far we understand from different kind of meeting and also different kind of study, yes, there is an effort to I would not say fill the gender gap, but try to increase the improve the imbalance of the women to work in the sector. And I think you can also see that, like for example, from through the forest education program, etc. But I think the regulatory effort and political is one side. Another side is how you perceive when women work in the sector as well. So I think this is depend very much on the type of work and how the woman perceive and how the sector itself perceive that the women would like to work in the sector. So I think all these things would combine and help to narrow the gender gap. I don't think that I answer the question completely, but I hope I can motivate something further.
Yonca, do you want to add something on that?
No, I think Rattiya covered the answer because also neither myself I would be able to answer this question, but Rattiya's answer is good enough. Thank you.
Okay. All right. We have one question. We have a few minutes or a minute to entertain one more question. Is there any opportunity for women to build up a separate community in this sector? Anybody?
Maybe I think for the separate community, I think maybe the audience can answer that better than me. But from my opinion view, I think yes and no. I still believe something like if man and woman work together, we will have a great how you call opportunity to move forward to something better. But of course, I think there is an opportunity for women to build up a separate community in the sector. For example, in many of the African region where I work, there's a lot of female entrepreneur, the female entrepreneurship in the wood-based sector. And they are very successful. And I think this is already shown that women can build a separate community. The second question on the context of Africa, yes, officially, the labor force survey would capture already the informal employment. But we can also assume that there might be some invisible number that the survey may not be able to capture. So I would say not 100%, and that's why we always say like the estimate is always the minimum number. Maybe my colleague Yonca can
No, Rattiya, I agree. That's exactly what I was also confirming. That's why we always say it's the minimum number that we are capturing now, and also, I mean, the informal employment is defined after we define first who is employed. So yes, informal employment as such, it is captured, but we may not capture everybody, particularly also those people who are not identified working in the forest sector as their primary job. So if they are doing this as an additional job or secondary job, we may not be able to capture these people. So that's the only thing. But yes, we are capturing at least part of the informal employment. Yes. Thank you.
Awesome. All right. I know there are still a few more questions in the chat box, so you guys could probably also answer them. But for the interest of time, we will proceed with the next speaker. All right. Thank you so much, Rattiya and Yonca. So our next speaker for this session is Dr. Iva Franic. She is a forest health researcher at the Swiss Federal Institute for Forest, Snow, and Landscape Research. She focuses on tree microbe interactions to improve biosecurity and forest health. And her current work delves on fungal transmission from seeds to seedlings, seed treatment efficacy, and RNA-based detection of viable seed-borne fungi. Dr. Iva, take it away.
Do you see my presentation?
Yes.
Yeah, okay, because I don't see it. Okay, cool. So good morning, everyone.
Excuse me, Eva. It's still in the power presentation mode. Yeah, there you go.
And
It's
now,
already. Yes. Yes.
is it
Thank
good
you.
now?
Yes.
I think my
Yes.
laptop is a bit slow. That's the problem.
Okay.
But okay, it's good now. Thank you. So yeah, good morning, everyone, and thank you for being here for this presentation today. Also, thank you, Jessa, for a nice introduction. So I don't need to actually tell you anything about the first slide, and I can just move on. So before I start talking about microbiome and how it changed our perspective of plant or forest health, I would like to go a bit earlier in the beginning. So the first time that plant diseases were mentioned in history was around 300 years before Christ. So that was long, long time ago. However, then they were only mentioned. And then for 2000 years, nothing happened. So basically, diseases were considered to be God's punishment and to be basically a result of spontaneous generation. And this was until the mid-19th century when Henrik Anton de Bary realized that potato blight was actually caused by a pathogen. He named that pathogen Phytophthora infestans, and this was the birth of plant pathology. Around 100 years later, researchers realized that it's not only about a host and a pathogen, but actually the environment is also important for a disease to happen, and this was depicted in disease triangle, which was the basis for disease management from then on and still today. And then we thought we had it all figured out until in the 20th century, we discovered that trees do not live in isolation. They are in fact accompanied by the huge diversity of microorganisms that have potential to modulate the disease. And this is what I'm focusing on today. So what are these tree microbiota? So tree microbiota are mainly fungi and bacteria. There are some other groups such as viruses, but fungi and bacteria are mostly researched. They are highly diverse. They are abundant. So the recent study calculated that tree trunk, so trunk of a single tree, can host 1 trillion bacteria. So these are huge numbers of organisms. They are, as I said, highly diverse, and they are also specialized. So they are specialized by tree species, they are specialized by tree organ, there are different organisms living inside or outside of the tissues. So they are very, very complex communities. What is also characteristic for these communities is that they are largely unknown, which is not that surprising knowing that the field is relatively new. So many organisms, we don't know what they are. And if we know what they are, we don't know what they do. So there is a lot of things that still need to be discovered. However, we do know regarding their function that because they co-evolved with their host, there is a number of these members of microbiota that can help host adapt to stress or with disease or pest resistance. But we also do know that some of these organisms, when they're exposed to environmental stress, can shift from commensalic to pathogenic behaviors. So let's see how trees acquire their microbiota. So first way of getting microbes in is via so-called vertical transmission from the mother plant. So part of the microbes are through the seed transmitted from mother plant to the seedling. And then also trees can acquire their microbiota horizontally from the environment via wind, rain, insects, or soil. And this happens at all stages of seed to seedling or seed to tree development. So for a long time, researchers believed that vertical transmission is very rare in trees. And the reason for this was based mainly on research done on pathogens and it was based on a belief that tree structures are just too complex and change too much for microorganisms to navigate this. However, recent studies have been showing that vertical transmission may be much more important than we thought. And exactly that question, although quite not completely directly, we addressed in this study that I'm presenting here. So basically, we looked into what is the relative importance of host versus environment, or in other words, vertical versus horizontal transmission in informing tree seed microbiota. And for this, we looked at several tree species belonging to several genera and gymnosperms that we collected in botanical gardens across Europe and North America. And what we have shown here is that both host identity and site, so the environment, play important roles in shaping these communities, but host identity explains twice as much variation as the environment. And indeed, that can be seen in this heat map where you see these two main groups of red dots, which basically show this differentiation in fungal communities between different hosts, gymnosperm and angiosperms, and then if you look more carefully also between genera. So what our results also showed is that although some environmental variables were important, such as climate, congeneric tree species richness and native or non-native status of a tree species was not important, emphasizing again that at the stage of a seed, really the main driver of diversity is host identity and not the environment, indicating that vertical transmission might be actually happening, so that a part of this microbiota is coming from a mother tree. And this was recently confirmed. So by now we do know that for certain tree species, so it's not large scale, but for several tree species, we know that a large portion of microbiota that is present in seeds also will be transmitted to the seedlings. And then during the time in seedlings that are exposed to the environment, they will be mixing with whatever is acquired horizontally from soil, from air. With time, we do know that these communities shift from being quite opportunistic and generalist to being quite specific. And this is based on studies where we look at communities associated, for example, with wood, bar, and bark. And we know that these are specific. For example, with tissues such as leaves, this recruitment happens yearly, so annually, and their communities may vary much more and may be much more influenced by what is coming from the outside. Okay, so when we have the adult tree, so we were also interested what factors will shape the communities in adult trees, and for this we did this quite large-scale study with many tree genera in, I think it was 6 continents. This was also done in botanical gardens, and we collected dormant tree twigs. So we looked into twigs, buds, and leaves or needles in case of evergreen trees. We pulled those, and then we did this popular high-throughput sequencing. So we looked molecularly into organisms that are present inside. And what we have shown is that 3 main groups of factors shape these communities, and all fungal guilds, so saprotrophs, symbiotrophs, and plant pathogens. So their communities are shaped by geographic distance between location, by annual temperature mostly, and by how closely related their hosts are. And here you can see that these relationships are not always linear. So for example, maximum difference between 2 communities is already reached at a continental level. So within the continent, and then they are very different also across continents. And for example, main shifts in communities happen on high temperatures, so much more on high than on low temperatures. So what does this mean? So for us and for forest health. So we know that the number of invasive pathogens and the damage they pose has been increasing rapidly, especially in the last 50 years. And this is due to trade in plants for planting. So to mitigate this risk, we do need to understand the diversity of organisms that are potentially being moved with trees. And also by understanding the factors that influence their communities, this can help assessing the risk of their introduction, establishment, and spread in the new environment. So for example, seeds and dormant twigs that I've been working on are considered as low-risk pathways in comparison with seedlings with, for example, leaves or soil attached. What we showed here is that even these low-risk pathways contain a high diversity of microorganisms, some of which are potential pathogens and might emerge as problematic in new areas. What we have also shown is that geographic separation within and among continents means that communities are very different. So if we continue moving things, we will eventually introduce new pathogens. And this is visible, for example, from this plot that shows the origin of invasive pests in Europe, pathogens in Europe. So you can see that historically North America has been very important, but Asia is catching up. And this is the result of high diversity of organisms of Asia, of different communities that we have, but also about this huge pool of unexchanged species. And this is the way, probably the other way around as well. So, I mean, with new trade connections, we are for sure moving more things. What we also show with this, so high host specificity of our communities, means that if pathogens are introduced, their establishment will depend on availability of susceptible hosts. And finally, we do know that with climate change, these communities will change. We are not sure yet how, but the change is potentially problematic here. And this will happen directly, but also indirectly through change of distribution and susceptibility of hosts. But what when the pathogen is already here? So what when we have a disease? So basically what microbiota can in this case help us with is that it can changes in microbiota in trees that are infected by either newly arrived pathogens or that emerge as a result of environmental change that triggers a disease. So changes in microbiota can tell us if something is going on. So we will for sure see the shift in disease trees, but what we can do is actually to look into trees that in presence of a disease are not symptomatic, and then try to find a solution in those trees because they might have organisms that are helping tree to be better. And for example, we are having such a study now going on in our group where we are looking into a very well-known pathogen, Cryphonectria parasitica, which is non-native to Europe and quite damaging for chestnut trees. However, this disease is very well controlled by a virus, so hypovirulent, that causes hypovirulency. But what we do not know if there are any other organisms that may help mitigate the disease. So we are currently looking into bacteria, fungi, and other viruses to try to understand if these passive cankers, so cankers that are healing, are actually, if the hypovirulence is supported by some other organisms that are helping the tree. And with this, I would like to just quickly summarize so what we know and where we go. So as I mentioned already in the presentation, what we do know is that microbiome communities are very diverse, and we do understand more and more how they are structured and what structures them. What we do not know is what are the mechanisms that are behind this structuring and what are their functions. The second problem is this huge proportion of unidentified species. many of us are moving more and more towards molecular methods to describe these communities. We are facing the problem of not knowing what the species are. And although there is a debate, but it is likely that we will need to go back to culturing, that we will need to describe new species and give them names so we can actually understand what's going on in these communities. Another thing is that this culturing is very important because we need to understand what are the lifestyles and functions of this microbiota, and this we can only do, or not only, but this is well done by actually inoculation tests and all kinds of experimental work. What we are also considering is moving from DNA to RNA research and metatranscriptomics to try to describe this function and interaction better. And finally, once we figure all that out, we will be able, hopefully, to start designing synthetic microbial communities and help forest management by these tiny creatures that live in the trees. And with that, I would like to thank you for your attention. I would like to thank our funders and everyone who contributed to this. And this is the picture of phytopathology group at VSL. And we are very, very happy to host people and to collaborate. So let us know and write to us if you're interested. Thank you.
Awesome. Thank you, Iva. All right. Yeah, we now have time for questions from the audience. And I think we have one here in the chat box. All right. Do you think then that assisted migration suggested often in the face of climate change is risky? And what are the alternatives?
Yeah, I do think, I mean, of course, yeah, it is risky. Assisted migration is something very interesting because it's going on and it's quite developed and there are people doing it. And every time we talk to those people, including at VSL at our institute, they're not very well aware that when we move things, we move also other organisms. Luckily, this is happening mostly on a smaller geographic scale. So, for example, we are moving things within Europe. So within Europe, we anyway are allowed to move things because we have the same trade. But however, what is really interesting, there are laws on how you can move, laws and advices how you move seeds depending on the climate zones. And that already helps. So it's not driven by phytosanitary motivation, but it already helps because we are not moving things too much. However, as I said, I mean, as long as we are moving native tree species and within a certain smaller area is not as bad as moving things intercontinentally, but it is risky. Yeah.
Awesome. All right. I think we no longer have time for another question, but if you guys have questions if there are questions from the audience, please do type it in our chat box, and we could have Iva answer them later for you. Okay. So now we will take a 5-minute break. All right. So we're back. Welcome to the second half of session 1 of the IUFRO webinar series on women in science. And our third speaker is Shae Swanepoel. She is a PhD candidate at the University of Pretoria in South Africa, studying eucalyptus defense mechanisms using quantitative genetics and functional genomics. Her study aims to uncover the molecular drivers of disease resistance to support the development of more resilient eucalyptus plantations. Shae, over to you. Oh. Mm-hmm. Shae? Can you hear us, Shae? I think your audio isn't working.
Hello, can you hear me? Oh, perfect.
Yes.
So I think I was having some sound issues, so I hope my introduction was good. Okay, I'm going to share my screen. Can you see?
Yep.
Okay,
Yes, we can.
perfect. Thank you. Thank you so much. Okay, apologies for that. My name is Shae Cooper. I recently got married, so I still go by Shae Swanepoel professionally. So if you see interchangeably Shae Cooper, Shae Swanepoel. But I'm a PhD student and I work under the supervision of Professor Sinushka Nadi at the University of Pretoria. And in this research group, we primarily focus on defense mechanisms or the molecular mechanisms that underlie disease resistance or susceptibility in eucalyptus to different biotic challenges. And so this study that I'm currently undertaking is looking at combining the data that we've generated through the years in a co-expression network to identify common defense responses of eucalyptus, and so that these genes that we identify can be prioritized for future studies. So just a brief background on forestry in South Africa. It's estimated that there's around 1.2 million hectares of timber plantations in South Africa, and there are three primary genera, which includes eucalyptus, wattle, eucalyptus, which takes up around 43% of the plantations, pine, which is around 50%, wattle, which is around 7%, and then this little small gray bar, which is consisting of other species, which is around 0.4%. But Forestry South Africa estimates that around 27% of the annual harvest is lost to pests and pathogens, which equates to around 938 million rand. And that's a significant monetary loss that we experience due to these threats. And so in South Africa, hardwood species are primarily used for pulpwood and mining timber, and eucalyptus contributes around 43% of this. And one of the commonly planted eucalyptus species is Eucalyptus grandis, which forms the basis of the studies that we perform in our research group. So the threats to eucalyptus, there's many threats to eucalyptus, of course, but what we primarily focus on in our research group includes Austratoxina acidii. So this is commonly referred to as myrtle rust, and it forms these striking yellow pustules on susceptible leaves and shoots. It causes defoliation, and in extreme cases, it can also lead to plant death. So in South Africa, Austratoxina acidii has not caused significant issues in our forestry plantations. We mostly see infections in more of the native species that are ornamental rather than in our forestry industry. So it's not currently an issue, but with the disease severity and the emergence of this pathogen as a global pandemic, we really need to start thinking about ways to mitigate more introductions of this pathogen into the country, and also how we can mitigate the losses that this pathogen causes. We also focus on Crassipulsi osteoafricanus. So this is a stem canker pathogen. It causes these necrotic stem lesions, and the plants tend to girdle. And in severe cases, again, it can also lead to plant death. We also focus on Leptospira invasiva, which is a blue gum culsid. It's a pest rather than a fungus, and it forms these galls on the surface of the leaves, and it tends to make the leaves sort of fold over on themselves, and then as a result, we see plant death as well, as the photosynthetic properties of the plant are altered. We also have Phytophthora cinnamomai that we focus on, and this is root rots. It causes, as the name suggests, root rots, and it's also quite a bit of a problem in our industry as well. And then finally, we also have transfer destruction, which causes shoot and leaf blight, and again, in very extreme cases, we do see plant death. And particularly in our forestry industries, we are noting an increase in transfer disruptants infections in field. And so what my questions are for my research was, what are the key genes that are involved in defense? And so from previous studies, particularly this Nupta et al. Paper, they observed that in Arabidopsis, pathogens from different kingdoms deploy these virulence proteins that interact with a limited set of highly connected cellular hubs in the host. And so what this means is then that there's a limited set of genes or proteins within the host that pathogens from different parts, different kingdoms target. And so this leads us to believe that there are then these conserved defense mechanisms that we can potentially exploit in our eucalyptus species. And to add to this, in an earlier study that our research group conducted, they looked at combining these data sets that we have available for eucalyptus to identify a core set of genes that are involved in either resistance or susceptibility in eucalyptus. And they identified that a range of different genes that could potentially be conferring this broad spectrum disease resistance. And so what this all comes down to then is that we need a more comprehensive analysis to unravel the complex interactions and to identify key genes in eucalyptus defenses that we can then prioritize for future functional studies and then potentially implement them into controlled breeding programs way in the future. So let me just quickly go over a co-expression network, what it is and why it is so powerful. So a co-expression network takes the expression of genes within your system. So for us, it would be eucalyptus, and it correlates the expression of one gene to the expression of another gene, and it forms this huge network of how these genes are correlated to one another. So gene A to gene B to gene A, etc. And through this, you can then construct a network of how these genes, based on their expression, are interacting. So here you'll have the node, which is the gene, and the edge, which is this bit here, and how that correlates then to that gene. And so from this, you can kind of pull different modules of co-expressed genes and infer function of those co-expressed genes, because you can infer that if a gene is correlated with another gene, it may have a similar biological function. And through that, you can then identify hub genes. So these are genes that are either intramodular, which are hub genes between modules, or intramodular hub genes within modules. So genes that are controlling the expression of that module. So think of it as potentially a transcription factor that is responsible for controlling the expression of genes within that module. And then you can also get intramodular hub genes, which are genes that are controlling the expression of the different modules. And so our then question was, what are these limited sets of highly connected cellular hubs in eucalyptus? Because the previous study showed that there is this set in Arabidopsis, so we wanted to know what is happening in eucalyptus. So then the aims of this research then were to investigate the overlap in the gene expression across the different pest and pathogen responses that we have observed within our research group, identify potential hub genes involved in these responses, and then determine the metabolic pathways associated with these genes. So I've got a list here of the objectives. We first wanted to identify the eucalyptus transcriptome datasets to use in the study. So as I mentioned before, in our group, we've studied Chrysoporthy, Ostrea africana, Leptocybe, Phytophthora, and Gyrosperia. And from this, we have around 180 different transcriptomic data sets or RNA-seq data sets that we have available to integrate into this co-expression network. And this consists of either mock-inoculated or inoculated treatments. And then there's also a data set with uninoculated treatments. and then there's a component of resistant and susceptible hosts at different time points. So it's quite a heterogeneous set of data that we used for this. So then we wanted to map these transcripts to the reference genome and quantify the gene expression. So I won't go into detail on that, but I rather wanted to show the differentially regulated genes. So we identified genes that showed either upregulation or downregulation in comparison to the controls between the different pathosystems that we used. So we then combined these to see where the overlap with the differentially expressed genes were, and we identified 587 genes that could potentially unveil or reveal the biology of the systems that we were looking at. And from these 587 genes, we performed gene ontology enrichment. So this is looking for gene ontologies or functions that could be related to the genes within this 587 sets. And we saw that a lot of terms associated with salicylic acid signaling were coming up, responses to nitrate and nitrate transport as well, which is interesting because in the previous study that our research group performed, they also observed nitrate as essential to defense responses. And so, like I say, this was shared amongst the differentially expressed genes. I then wanted to look at the co-expression network and interrogate the genes of interest that come out of that network. And so we used a WGCNA package in R to construct a weighted gene co-expression network analysis to identify these modules that are strongly correlated with one another, or the expression rather is strongly correlated with one another. We identified 38 modules and hub genes that had significance within these modules. And we also then looked at what the expression of these genes in the modules looked like when we compared it to the set of differentially expressed genes. So were they responsible for defense responses in the different pathosystems, and what did they do when they were in the co-expression module? And then again, we performed functional enrichment of these to see which biological pathways were typically enriched. And so I've got this heat map here. It's a little bit complicated, but what I want you to focus on is that we've got three separate divisions for the different samples that we had. So we had mock-inoculated, inoculated, and uninoculated samples. And then the host, no, not the host response. For this particular one, what I want you to take note of is that these genes are down-regulated, or expression is lower in the mock-inoculated compared to the inoculated samples. And you can observe this with the module eigengene, which is just the representative gene expression for that module. So in general, before inoculation, the expression of these genes was quite low. And then after inoculation, expression of the genes typically tended to increase per the different pathosystems. And so this is what we're considering a core defense module, because it is the expression of these genes are being induced upon inoculation with the different pathosystems that we have. And so when we looked at what the enriched genes were within this module, we found again that nitrate transport and responses to nitrate were enriched, as well as salicylic acid signaling, among other common defense responses that are typically observed in our data sets. So again, it just included salicylic acid and nitrate transport. So nitrate transport, we then wanted to investigate what the different nitrate transport genes were doing in our different data sets. So I've clustered these as resistant and susceptible samples, and then the different nitrate genes that include uptake, xylem loading, phloem loading, and vacuole storage. So typically, transcriptionally, the nitrate transport genes appear to be differentially regulated, which means that upon inoculation with the pathosystem or the pathogen, there is a transcriptional response from these genes. So these genes are doing something. We're just not entirely sure what it is that they are doing. In general, you can see that they are down-regulated in resistant hosts, and then somewhat more upregulated in susceptible hosts. So is it potentially that the resistant hosts are switching these genes off during infection to prioritize growth responses? And this is something that our research group is exploring further. Then I wanted to show you a growth defense trade-off module. So again, we have the mock-inoculated, inoculated, and uninoculated samples. And before inoculation, the expression in general of these genes is significantly upregulated, or higher rather. And then after inoculation, there does appear to be a decrease in expression of these genes. And so when we look at what the genes are within this module, we see that it's genes that are more involved in growth than defense, and that includes things like root hair elongation, sepal formation, and meristem growth. And so from this, we hypothesizing that there's a set of genes that are involved in the growth that are being switched off when being inoculated, to suggest that there is this growth defense trade-off. And that there is a difference between, I don't think I mentioned that, there is a difference between resistant hosts and susceptible hosts, where susceptible hosts appear to not be able to switch these genes off as well as resistant hosts, potentially leading to their susceptibility. So if we look then at a model of salicylic acid and nitrogen in plant defenses, so this is something that we are hypothesizing. So what we know from this model is that when there's an infection by a pathogen, it, among other things, activates salicylic acid signaling, which leads to downstream activation of what's known as pathogenesis genes. And these then lead to systemic acquired resistance and local defenses. And this is the typical model that we normally see. Then in a study that was performed in 2012 by Jimenez et al., they looked at how nitrate transport genes influence this pathway. And so what they found was that when they switched off the expression of NRT, which is involved in nitrate uptake, that then gets fed into this nitrate assimilation pathway to lead to acquisition of nutrients. When they switch this gene off, it's led to impaired nitrogen metabolism, or they impaired the nitrogen metabolism, and this resulted in rapid salicylic acid priming, which resulted in increased resistance within Arabidopsis that they were studying. And sorry, my keyboard's not working. And then there's also then reduced pathogen proliferation with this reduced nitrate uptake, because the pathogen cannot acquire the plant's nutrients. And so what we are hypothesizing then is that these NRT genes that we are observing are potentially contributing to plants' susceptibility in a sense, because this salicylic acid pathway is then interfered with, and we don't see this rapid priming of defenses for the different pathogens that we're observing. And so, like I say, impaired nitrogen metabolism induces this rapid salicylic acid priming, which leads to increased resistance and less nutrients for pathogen proliferation. So when the genes are knocked out, there's this increased resistance. And so we wanted to know then what happens if we do knock these genes out in our systems. But that's, again, a future study that's not a part of my research. So then, just to conclude, we identified defense core modules that are induced upon infection. Nitrate transport appeared to be crucial in defense, and salicylic acid signaling also were triggered in response to infection. And then the susceptible hosts were enriched with genes involved in cellular processes. So there's the theory that they then cannot adequately modulate between growth and defense, and focus is diverted away from defense rather than for enhanced resistance. And so looking forward, this is where research in our group is going. So we've performed now these eucalyptus defense studies and identified this pathway as potentially important in eucalyptus resistance. We've identified hub genes, and we're moving towards functional genetic studies, not necessarily in eucalyptus, because to transform eucalyptus is quite difficult to do. But in our group, we are looking at what these NRT genes do and what knocking them out does to the transcriptomes of model plants like Arabidopsis. And then we're also looking at that in poplar to see if there is actually a link between knocking these genes out and increased resistance in a hardwood species such as poplar. And then with that, I'd just like to thank the research group and all my funding bodies. Thank you.
All right, thank you, Shae. All right, time for questions. Do we have any questions from the audience? We have one here in our chat box. I might have already talked about this, but what are the cellular functions of salicylic acid and nitrate pathways? And do you think there will be crosstalks between these?
Yes, yeah. So for salicylic acid, what it tends to do is it leads to I think I did mention this, the expression of genes that are involved in defenses, which is these pathogenesis-related proteins. And through that, it leads to systemic responses of disease resistance. There are other functions of salicylic acid. It's not necessarily just defense, but we obviously focus on it in the context of defense. And then the nitrate pathways, they are for nitrate uptake from the soil. So the plant will take these nitrates up and then assimilate them into pathways for such as amino acid production. So it's very growth related. And I do think that there is a crosstalk there. There have been studies that show it may be not a direct crosstalk. So in terms of disease resistance. So I'm not sure what the actual role would be between salicylic acid and nitrates, but the literature tends to suggest that there is a link there. So we do need to look into that a bit further.
Awesome. Okay, but really quick question. There's one question uploaded. How do the results of this study aid in breeding programs for eucalyptus species? Really quick answer to that.
This is a good question. So it won't directly, I think at this stage in our research, it won't directly aid in breeding programs. That would come way downstream of studies like this. For this study, it's very hypothesis generating. We are generating hypotheses that these genes are involved in resistance. And then through downstream functional studies that can show that these genes are involved in resistance, we could potentially better guide breeding programs to ensure that they incorporate species or varieties or whatever the case is that better express those genes, for example. So for this particular study, it's breeding programs is not really what we focus on. It's more, like I said, hypothesis generating. But that's a good question.
Awesome. Okay. There is one more question, but I don't think we would have time, but maybe Shae, you could answer this question from Pascal. All right. Okay. Thank you so much, Shae. And we will now proceed with the fourth and last, but definitely not the least speaker for today, Priscilla Kimani. She is a research biotechnologist at Kenya Forestry Research Institute and currently pursuing a PhD in genetics and plant breeding. She has worked on projects supporting commercial forestry in arid regions, focusing on tree breeding and improvement, tissue culture and micropropagation, and assessment of genetic diversity of plants. Priscilla, we're so excited to hear from you. Please begin.
Thank you very much. Is my presentation visible now?
Yes.
Okay. So thank you very much for the opportunity and welcome to the presentation. My presentation is on tissue culture of woody plants, focusing on the achievements, opportunities, and constraints for Africa. So this is basically a review of where we are and what we are anticipating that we could do with tissue culture of woody plants. So just to give a brief background and introduction of what is plant tissue culture. So tissue culture is basically the regeneration or the growing of plants from small parts of the plant. You grow whole plants using any part of the plant. So the part of the plant could be the leaves, you could use the roots, you could use the stems or the buds, and even isolated cells. And we do this in aseptic conditions in a very clean environment, and we are able to just, instead of just using the seeds as is normally done to regenerate the plants, we use any other alternative parts. So the applications of plant tissue culture include the rapid propagation of germplasm. And so we are able to multiply. If you have a small stock or a limited stock of probably improved material or bred material, then we are able to multiply them very fast. We also have clonal production of maybe selected germplasm, and this one is very useful in breeding programs. So we might have isolated or selected some certain plants that are very unique, and we want to do clonal production, and the tissue culture comes in very handy. We also do callus culture used for the synthesis of secondary metabolites, and which is also an alternative to the traditional ways of isolation of secondary metabolites. Tissue culture is also used for genetic transformation, somatic embryogenesis, which is also used for mass production, and even cryopreservation of germplasm, especially when I'm talking about endangered species. So because we're talking about the woody plants of Africa, and basically this includes the trees, it includes even fruit trees, and why they're very important. These species have been used for a long time in international trade. So we all know the mahoganies of Africa. We know unique species like Dalbergia melanoxylon, the hardest timber in the world. We have other species like Melissia species, which are also world renowned for good timber. They also provide us with non-wood products like the bioactive compounds that are used in pharmaceuticals. They have been used for human medicine, for even veterinary medicine. We have agricultural uses like biopesticides, especially in this era, where we are trying to reduce the use of synthetic compounds. They're used in cosmetics. And also importantly, we have the fruit trees falling in this category, and they provide us with natural fruits. They have also other services like aesthetic services, ecological services, where they are used for habitats of wildlife. And this also helps us to, in services like tourism, we are able to conserve the environment, and also the environmental services like holding of the soil, preventing of soil erosion, and climate change mitigation. Other issues and other uses are issues of socioeconomics and the provision of livelihoods for many families and communities in the African context. So why we go for tissue culture of woody plants? So number one, if you're talking about trees, we know that they have many of them, especially the tropical trees. They are prone to very slow natural regeneration. They grow very slowly. For a tree, or for many of them to reach even reproductive maturity, they take a while. They will take between even 5 years, 10 years, that's even 20 years. And after they reach the maturity period, they are also now going to produce the seed, but the seed production may also take a while. So for many species, you'll find they will take a year or 2 years to just grow the seed. So we also have very many issues with seed propagation, and the seeds, most of them, because the tree is really trying to protect its seed, and sometimes the conditions are not very good for the germination of the seed. So there is a lot of seed dormancy, and to break the dormancy, also we end up destroying many of them. There is low viability of the seeds. There is poor storability. You try to store them, and maybe also because of the infrastructure, they require some certain temperature, but we are not able to reach it, so they will spoil, and we are not able to grow them later. So we end up with low germination rates, and it becomes a challenge now to sustain our production or our plantations. Again, we also have limited success with vegetative micropropagation. So this means when we want to probably root cuttings of certain trees, they are very recalcitrant. So we are not able to do that very easily. And again, that is also space intensive. It is time intensive and also very expensive. And again, at the moment, we have increased demand for tree products and services in the continent, and of course, even worldwide, because we have increasing populations of people, we have increased urbanization, so even the available land is decreasing. And so we need really to look for alternative ways of increasing our production of forest products. So some of the tissue culture methods that are used to produce these woody plants, and of course other plants, is direct organogenesis. What this means is that you directly initiate shoots, and then we are able to multiply them, eventually rooting them. Again, we have calogenesis and somatic embryogenesis. So in this particular presentation, I would like to give a case study of the tree species that we have really focused on in terms of research of tissue culture. And we have a species of tree called Melia volkensii. This is an East African dryland mahogany that is basically its common terminology. This species of tree is drought tolerant. So especially in our Kenyan context, we have very vast drylands. So drought tolerance is very important for us when it comes to trees. We also have issues of, so where we have vast drylands, then there are also challenges of termites. The termites will attack the trees and they are not able to survive. So the tree is known to be termite resistant. It is also pest resistant, or rather resistant to many pests, which is also a challenge when we're raising trees in the drylands. It is a popular tree with farmers and also the communities where it grows. And we have an improvement program. So this tree was recruited into an improvement program, a breeding and improvement program, about the early 2000s. And so far, we have quite some advanced generations of improved genotypes. So what that means is that we have been using conventional means of propagation, but where tissue culture comes in is, as we are giving examples of direct organogenesis. Direct organogenesis, as is indicated here, is whereby we initiate some shoots and then directly they're able to propagate themselves into many and multiple shoots. As it goes around, you see that they're able to now come and root, and you have a tree. You have a micro tree there. So what happens is that once you start with a small shoot, then you're able to get very many plantlets out of one. And that becomes a lot easier. So when you're talking about probably the breeding program of Melia volkensii, we have a way of multiplying many of the improved genotypes, and that becomes a bigger advantage where farmers are concerned. So another case study of the same tree, is embryo culture, and this also has been a subject of study where we were able to propagate or to extract embryos at a very early age. And why that is important is because Melia volkensii, as a case example, it takes about 13 months to mature the seed. So in the context of a breeding program, that means that once we plant one generation, then we have to wait for a whole year or more than 1 year to be able to harvest the seeds and advance it to the next generation. So this becomes important. Embryo culture, this was done by extracting the embryo from an immature fruit. And from that point, it was able to be regenerated into a whole plant. And this happened about when the fruit was about 6 weeks old. So that means that you can shorten the breeding time by almost half a year or thereabout. So that is also, there's some more applications, including the embryo rescue, which means that if you have interspecific or intergeneric hybridization, and some of these embryos do not see maturity or they do not grow to maturity, then we're able to extract them very early, and we are able to grow them in vitro, and they survive into whole plants. So the another importance of embryo culture is circumventing seed dormancy, which I had mentioned that is a big problem when we are growing woody plants. And of course, also shortening breeding cycles, and even the use of genetic manipulation to improve plants. So some other work that has been done on the tissue culture of Melia volkensi is somatic embryogenesis, and we have been able to grow or to generate somatic embryos from a very early age of the plants. And this is very important because it can help with the rapid and large-scale propagation, because with somatic embryogenesis, you generate very many somatic embryos, and these you're able to mature them, and they're able to grow into normal plants. So it is also useful for clonal production, where you have selected germplasm or improved germplasm, and we grow them into many plantations. We can also do this for synthetic seed. We can produce synthetic seed, which is now able to be preserved probably for a longer time and even transported a bit more easily than the seedlings that are already grown. Sometimes they really are destroyed during transport. So some other work that has been done for Melia volkensi, through in vitro manipulation. Some studies have reported the production of tetraploid genotypes, which I think are still continuing to be tested, but initial reports have shown that they are more compact, they have thicker stems, they're wider leaves, and even probably can survive drought, even war, when they have some other physiological features. There is also genetic transformation of Melia volkensi that has been attempted and even published. Now, those are not, so that was just a case study of Melia volkensi, but that is not the only work that has been done. And we have done a background of more examples, which you can probably just look at later. It is a chapter called Tissue Culture of Threatened African Hardwood Tree Species. And if you search that, maybe you can be able to look at more examples of what has been done. So what are the opportunities for Africa as far as tissue culture is concerned? So because of what we had said, the lengthy growing periods of the trees, then there is potential for acceleration of breeding. If you look at the case study of Melia volkensi, we started already in the 2000s. That's about 25 years of breeding, but the most of the time is spent waiting for seed. It's spent waiting for the conditions to be right, even for the seeds to be grown and in the field. So there is potential for accelerated breeding, especially now when improved germplasm is really in high demand. There is also the potential for plantations of the high-value woody plants, and that will eventually improve the livelihoods of the communities, as well as enhance the trade for these internationally traded timber. There is the potential for genetic conservation of the endangered species, because, of course, with the logging, with the deforestation, we have been losing quite a number of species, and this tissue culture can be very important for this. and also to support the ongoing reforestation programs aimed at mitigating climate change. Some constraints and challenges that are faced in this field include that most of the tissue culture programs, if you look at publications in the African context, you find that they're at the developmental stages. So there's really a lot to be done. There's, vis-a-vis other countries, you will find that we still have a lot to do. There is the lack of standardized or uniform methods and protocols for most of these species. So you find different authors are reporting different methods for the same species. So maybe we could just have standardized methods. The methods that are developed are not usually upscaled. We just leave them, they're very basic, basically. So just to upscale them to a level of being able to use them commercially. Of course, the issue of limited funding to research, basically and generally, and infrastructural constraints in terms of equipment, in terms of capacities also, and also very sometimes the policy is not very supportive. So some recommendations include enhanced capacity building and training for those interested and for the experts to build expertise. Public-private partnerships for upscaling commercial application because there is potential. There is most of the examples that exist have shown that it is possible to actually grow these woody plants through tissue culture, but we need upscaling to now the commercial application aspects. And we also have increased funding. We would recommend increased funding for research and application, and again, to develop government policies that support the environment for conducting successful tissue culture. Again, regional and international collaboration, which we are also very open to. And that is my presentation. Thank you very much.
Awesome. Thank you, Priscilla. That was a great talk, and we now invite questions from the audience. We have here one question. I wonder how tissue culture is received or perceived by tree farmers, and are these freely available for them to use?
Yes, so the farmers are very receptive. One advantage of using tissue culture, especially where farmers are concerned, is that we are able to lower the price of the germplasm. So some trees, for us to grow them using seeds, we have to wait for the seeds. So by the time this waiting period, it translates to an increased price of the seedlings. So when we conduct tissue culture, then we're able, number one, not only to produce many of them, so there's definitely economies of scale. And again, we are also able to shorten the time. So the farmers are receptive. And again, the tissue culture plants grow as normally as the other plants. But the added advantage also is that we can do clonal production. So if we have elite trees, then we are able to maintain that morphology and give to the farmers what the best of the best. We reduce the genetic diversity of the seed.
Okay. All right, there's one more. I'm not sure if I missed, but are there any trials of your work in the field or in the near future for these tissue culture products that you did?
Yes, we have trials in the field. We have demonstration sites in the field from the tissue culture experiments, the plants that we grew from there. We have taken them to the field. We have also provided and supplied some to the farmers, so they do exist. They are also in commercial plantation, although on a small scale, not yet at a large scale.
Awesome. Okay, one more question. How do government policies influence scale up of tissue culture programs in African forestry?
All right. Basically, it's the issue of the fundings and the allocations to research in general, and especially where tissue culture is concerned. Tissue culture falls in the research first and foremost. So the support that is given to tissue to research, the funding that is allocated, to the research. It's basically where the priorities are aligned, actually. So for us, we feel that the policies could align more to supporting research and also to supporting the upscaling of the product of tissue culture.
Awesome. Okay. Do we have some more questions from the audience for Priscilla? Okay. All right. Thank you, Priscilla. But we still have time for open discussion. So maybe if you have any questions from the other speakers as well, please type in the chat box so we could so our speakers could also answer them. All right. Okay. There is one here. Hi. Oh, this question is for Eva. Eva, I would like to ask, what are the factors for the latent microbes that you have mentioned previously to become pathogenic, and how will this translate to forest health management?
Yeah, thank you for the question. I think one of the main factors are environmental stresses such as drought. So for example, in Northern Europe, we do, okay. In the Northern Europe, we do have, I mean, everywhere in the world, but also in Northern Europe, outbreaks of diplodia, for example. So there is this whole group of microbes from Botryospheriaceae family that are endophytes, or they have endophytic stage, and then they can emerge as pathogens upon drought periods, but also like forest fires and all kinds of disturbances. How will this translate to forest health management? Well, yeah, I mean, I guess we will need to be aware of these things and just be prepared that, I mean, more we know, more we can be prepared for after these kind of events, that we have strategies that we can act immediately and not wait for outbreaks. Yeah.
Thank you, Eva. Let's see if there are some more questions that haven't been answered yet. I think there's one here from Julian Kabwe, and I think this is for our first presenters, Yonca and Rattiya. In the context of Africa, did you consider informal sector? Oh, okay. So I don't, oh, maybe they already, okay. All right. There is, oh, that was already answered. Okay. Thank you, Jillian, for that. Let's see. Do we have some more questions for our from our audience for our speakers? All right. Going once, going twice. All right. So I think oh, there's one more question for Priscilla from Pascal. Which are the traits targeted by tree breeding programs?
All right, so I think it also depends on the tree species that is in question, but maybe just to give the example of Melia volkensi. Melia volkensi it is a slow grower based on the fact that it grows in the drylands. But one trait is to improve it for fast growth so that it takes a shorter time for maturity or for harvestable maturity. So we have been able to reduce from around 25 years to 15 years for fast growth. There is also the diameter of the trunk, you know, how big the tree becomes, because our main focus is wood or timber. So we want biomass, and we want to improve for biomass. We want to have trees that are growing straight instead of crooked stem, because we want to harvest good timber. There is also the branching habit. So some trees will start branching very, very low, but we want a long trunk and the branches to start high up. So those are just some of the traits that we look for. And again, also we want to improve drought tolerance because we are experiencing longer and longer periods of dryness or of drought. And so we also look for drought tolerance traits.
Awesome. Okay. There is one more from Jillian. How much mentorship are you doing and the interest around this? And great presentation.
Well, all right. Thank you very much for that. We are trying. We're really trying to spread the word around. We have a whole team of tissue culture researchers around in Kefri, where I work. And again, we also have this regional, we have come together sometime as the regional scientists. That is, so we have some scientists in Uganda, we have some scientists in Ethiopia, with whom we were also learning some other techniques of tissue culture in bamboo. So we're not only working on specific African species, but we're trying to also diversify and expand because our landscapes are diverse also. We are learning, we are working with interested people. Yeah.
Okay, there's an additional or follow-up. What would be your comments about the nature of investment for this kind of work in African institutions?
Well, yes. So on a small scale, we have investments in small scale. We have received grants in various times. We have received support, financial support. We, of course, we acknowledge donors like the JICA. They have really, the Japanese Institute or the Japanese, the one that supports their donors, and they support us to really put up some infrastructure. So for some institutions, you find that they're well equipped, but for many of the labs, we don't have. But we are making strides. There is the investment includes basically the building of tissue culture labs, which include the cold rooms and probably the incubators, the buying of equipment, capital equipment like autoclaves and the clean benches, and we are moving slowly. So it's being done. It's actually being done slowly but surely.
Yeah,
and
thank you for
of course, I should also mention we would appreciate anyone that wants to come in and work with us.
Yeah. All right. Awesome. Yeah, more funding for more research, right?
More
All
funding,
right,
more training, and more exposure also would be
Yep.
Yep.
Yep. Awesome. Any other questions from the audience? Going once. Going twice. All right. Okay. Thank you so much, Priscilla, and thank you to all our speakers. There we have it. We have 5 women researchers doing incredible work in forest sciences. But before we conclude this session, we want to extend a heartfelt thank you to each of you, all the speakers and the audience, our tech team right here, for joining us today. Your participation truly makes these discussions special. As mentioned earlier, we are again inviting everybody to complete the survey about women in forest sciences with the link to the survey already uploaded in the chat box. And please don't forget, we still have 2 more sessions coming up in the IUFRO webinar series on women in science, and we can't wait to continue the conversation with you. We hope everyone has a wonderful rest of their day or night, wherever you are in the world. Thank you.